The process of transcription starts with the transcription factors, which are proteins, binding to the TATA box, which is part of the promoter region. Then RNA polymerase II comes in after the promoter region and unwinds the double helix of DNA. Then it will bring up pieces of RNA which match with the DNA, and it will go along and do this to the whole chain. Each codon, which is made up of 3 nucleotides, will match with a codon from DNA. Then the RNA strand will disassemble from the DNA, move down, and the DNA will reattach itself.
The next step is going to be translation. So before the RNA can leave the nucleus, it needs to gain a methyl G cap on the 5' end and a poly A tail on the 3' end. The poly A tail helps protect the RNA strand from enzymes that are trying to get to the codons. So the longer the tail, the safer the RNA will be. The next step that's initiated is called splicing, which is done by the spliceosomes, which have SNRPs on them, which will get the introns away. This will allow the exons to join back together and start coding for their anticodons. The next step in this process will be to bring up the small ribosomal subunit, followed by the large, with the AUG or start codon in the P site, which will bring over the anticodon and the matching amino acid over to the synthetase enzyme, where it will combine and be brought over to the codon. This will initiate the ribosome to move over, having this anticodon in the exit site, which will allow it to leave, and moving on to the next codon, which will be in the P site. The same thing will happen in the enzyme, attaching the amino acid, which will be brought over and attached. But now there's going to be a polypeptide bond caused by GTP in between the amino acids to form the polypeptide chain. And this will just continue to occur all the way down the chain. Bling. When the ribosomal subunit gets to the end codon, UAG, it's going to trigger a release factor to come over to the peptide chain and drop a water molecule, which will then allow the methyl G cap and the poly A tail to dissolve and fall away, and same with the ribosomal subunit.